All right, welcome back, everyone. We're not going to start with a coot. We're going to end with a coot. I hope that you survived, if you're local, this crazy weather. My partner went for a run right before this class started and got hailed on. Okay, so we are going to pick up where we left off last time. And we had started this activity, or we were about to do a breakout. Before we do the breakout, I'm going to have you do sort of an individual practice. And what I'd like you to do is look at the prompts on your screen. These are 6 different scenarios. What if you wanted to look at the various different things? I want you to think about which microscopes you would use and what your reasoning would be for using them. Don't put them in the chat. Just take a few minutes to do this on your own, thinking about it. And then I'm going to put you in groups to talk about it. All right, welcome back, everyone. So we're just going to have you do a raise your hand for this one. So you hopefully talked about it with people in your group and got some ideas. I'm going to go through each scenario, and quickly, I just want a couple of people to raise their hand and tell us what microscope they'd use and why. So let's start with number 1. If you wanted to determine if bacteria are swimming, which microscope would you use and why? Raise your hand if you found an answer in your group or by yourself. Sarah, go ahead.
I thought it would maybe just be just like a bright field microscope, because the specimen is able to be alive and able to move around when you're looking at it. So that's what maybe I was thinking.
Great. And does anyone have anything else that they want? I know that there was one other group that did this one. Does anyone or anyone else in your group have anything they want to add? Okay. Great. So yeah, you definitely want to use one of the microscopes that would allow you to see live specimens. And so that's going to definitely rule out electron microscopes. And it is possible to see bacteria swimming in the bright field, although it's quite tricky because of the contrast issue. There is a better option. What is that, guys? Put it in the chat if you can think of one. Kelly asked my cat's name. It is Pumpkin Jack. Dark field, yes, that would be a good one. I would say that would be the better option, although there are ways to set up your bright field microscope to see bacteria swimming, and I could do that with you in the lab. It's just more challenging. Okay, what about number 2? Did anyone come up with an answer for number 2? Raise your hand. Tell us what you chose and what your reason was. So for number 2, we want to see the detailed structure of bacterial flagella. This one does not require us to be alive or not alive, but we do need pretty high resolution, right? Certainly a light microscope's not going to cut it. So any thoughts in the chat as to which one you would choose? Great. And I'm just going to ask someone from the chat to tell me what their reason was for choosing that. So I'm going to go to Ayumi. What was your reason for choosing electron microscope?
I don't know how to say, but like you can see the very details.
Yeah, the resolution of it is excellent. Yes, I agree.
You're like really, yeah, pleasure.
Yeah, it might be the only one that would allow us to see that level of detail. Good. All right. Number 3 was map protein complexes on the surface of flagella. Did anyone come up with an option for that? Raise your hand. Megan, go ahead.
We decided on confocal because just for precision issues, because it is one of the more precise light microscopes you can use, and just because the flagella are so small.
So tiny, yeah.
Right.
Yeah. So it's interesting because this would actually work for a eukaryotic flagellum, and I didn't specify. So for a eukaryotic flagellum, certainly possible. Bacterial flagella are so teeny that you would have to sort of resort to probably an electron microscope again, or an atomic force microscope could do the trick too. All right, good. What about number 4? What if you wanted to know if a protein was part of the flagella motor? So this one's going to be like a co-localization question, like you know where the motor is. You want to know if there's a protein in it. That would be a co-localization experiment. Did anyone come up with an idea for that one? No brave souls want to raise their hand. What about in the chat? Put your answer into the chat if you had any idea for that at all. Yeah, I agree with you, Sarah. I think a fluorescence microscope might do the trick. Okay. It's tricky because, again, we don't know if we're talking about a eukaryotic or prokaryotic cell, and this would work for a eukaryotic flagella. In a prokaryotic flagella, it's iffy. You might have to do some kind of electron microscope thing because of the high level of resolution. What if you wanted to see the bacterial flagella moving, see its rotation? So this has to be live. Does anyone have an answer for that one? You can raise your hand or put it into the chat. I know it's been a day of hail. It's the end of the day. Everyone's like, I don't want to raise my hand. That's too much work. I'm too tired. So either raise your hand or put your answer into the chat if there was one that made sense to you. Well, so this one does specify bacterial flagella. We want to see it moving. It's going to be really tricky to use a regular light microscope, right? Because of the tiny size. Yeah, Haley, I would say a confocal would probably be your best bet because we want to see it moving, so it has to be alive. We need the highest possible resolution. Luckily, with a confocal, you get that good contrast, so it is actually possible to see bacterial flagella moving if you use the right conditions. Last prompt was number 6, visualize microbial community attached to a rock. So we don't want to disturb it, but we do want to image it. Put into the chat if there's one that seems like a good fit to you. Yeah. Yeah, I'd agree. Scanning acoustic would be a good option. Remember, scanning acoustic is good for getting like 3-dimensional images of stuff that's in situ, that is undisturbed in the natural environment. Okie dokie. So that was just kind of reviewing and trying to dredge up and apply our knowledge from last time. I wouldn't ask you such open-ended, vague questions necessarily on the test. The answers to those questions were not like, there wasn't just like one answer. It was more about trying to think about, what do I know about these microscopes? What are they good for? What would be a good choice? What would be a less good choice? Okay, so moving on, today we're going to primarily talk about the problem that microbes are transparent, and we're going to focus on the staining techniques used to visualize them. Here are all the ones you need to know, right? There's 3 major categories of stain: simple, differential, and special. Simple stain is very simple. You just add some dye, voila, it's colored. Differential stains are more complicated and they have a particular goal. They want to reveal something in particular. So the gram stain and the acid fast stain both reveal something about the cell wall of the organism. And then special stains are used to stain for particular structures that are hard to visualize by regular staining. Now, on a simple stain, you can see some basic things about bacteria. And really this part, just to remind us, is we're really focused on bacteria in this class. So we're going to really talk mostly today about that type of cell. When you're simple staining a bacterial cell, it reveals morphology and arrangement. So here are some pictures. I am going to animate through this with an arrow. And when you see the arrow pointing, I want you to tell me what morphology it is. Write that into the chat. So what morphology is shown by the cells there that the arrow is pointing to. Make sure to only answer for morphology, nothing else. Good. That's right. These are round, spherical. We might call them cocci, right? We might call them cocci. What about these guys? Yeah, I haven't lost my mind. I'm just making a point. These are also cocci. Their arrangement is different, but they're both have the same morphology. Okay, what's the morphology of these cells? Right. Correct. These are rod-shaped, also known as bacilli or bacillus. What about this morphology? Indeed. These are the fun ones. They are called vibrio. They have this little C shape to them, a little curve, a little bend. And these guys? Yeah, these are spiral, spirillum, spirilla. That's right. Mm-hmm. And last but not least, we have this guy. Now, you might think, oh, this one and that one are the same. It is not true. This one has a much tighter coil. And although you can't see it in this static image, it also, its motility has a corkscrew-like motility as well as a corkscrew-like shape, whereas this one has a looser curl, and it's often, if it's motile, has flagella to move it along. That's right, guys. This one is a spirochete. Good job. Now let's talk about arrangements. All of the ones that the arrows are pointing to have the same arrangement. What is it? What do we call it? When they're by themselves, we call it single. It's also fine to call it random single. And what are these? Correct. These are diplo. How about these guys? Yeah. Yeah, these are chained. You could also call them strepto. And what's this one? Mm-hmm. Yeah, clustered, also called staphylo. What about this one? Indeed, that is known as a tetrad. Okay, and this is cubical. That's right. Sometimes called sarcina. Mm-hmm. Yep, good. There's other ones. In the rods, there really isn't usually clustered rods, but there can be accordioned rods where they're kind of like zigzagged, and that's called palisades. All right, now that's what a simple stain will reveal. Differential stains tell you additional information. When you do a differential stain, you do see the cell shape, the morphology, you do see its organization, its arrangement, you do see those things. But the differential stain has another purpose, a major purpose. It's asking a question, usually has to do with the structure of the cell, perhaps the cell wall. So when you do a gram stain, what do you find out? Put that into the chat. What do you find out when you do a gram stain? You can also raise your hand if you'd rather talk it out. But if you can just articulate it in the chat, then we can read it out there. Yes, we find out if they have a thick or a thin cell wall. And we find out if they're gram positive or gram negative. Yep. Which one's which, by the way? The thick walled ones are positive or a negative? Positive, yeah. Now, when we get to chapter 4 next week, we're going to talk in more detail about what a gram stain organization, gram-positive cell wall versus gram-negative cell wall, what that really looks like. And you will have to know that detail. So what it comes down to is it's looking at whether the cell wall is thin or thick. And it has to do with the organization of peptidoglycan. Bacteria all have peptidoglycan, but how it's organized is different between the two types. The acid-fast stain reveals something different. What is it? Yes, Jenica, it is true that a cell that is considered acid-fast positive is resistant to normal staining. Okay, that's why we have to do the acid-fast stain. Sarah says whether or not the bacteria has a waxy wall. That's right, there's a chemical in the wall that is waxy in acid-fast bacteria. It's not present in other bacteria. What is the name of that chemical? Yes, Kelly, it is mycolic acid. That's right. So it is the presence or absence of mycolic acid. When mycolic acid is present, which is waxy chemical, then doing a gram stain or a simple stain is quite difficult because this wax repels normal dyes. So you have to do an acid-fast stain to reveal it. Okay, now there are reasons clinically that we care about these two stains. They're widely used for very specific and understandable reasons. Why would we care about the organization of peptidoglycan? Right? It tells you whether or not something's gram-positive or gram-negative. And if it is gram-positive or gram-negative, then we know that it is resistant or sensitive to what? We know that it is resistant or sensitive to, a couple of you have it. Let's see if everyone can get there. Yeah, resistant or sensitive to penicillin. Not resistant or sensitive to all antibiotics, but specifically resistant or sensitive to penicillin. And which of the types of organism, gram-positive or gram-negative, which one of them is resistant to penicillin? Is it gram-positives or is it gram-negatives? If you're not sure, you can just put a question mark, but do engage, partly so I know that you've answered, partly just so your brain is paying attention. That's right. It is gram-negatives that are resistant and gram-positives that are sensitive. And sometimes people get this backwards because gram-positives have a thick wall. And it's tempting to think that means it's more protective, but it's actually the opposite. And we'll find out why when we talk about the details. But gram staining reveals whether or not penicillin would be a good treatment. And gram-positive is sensitive to penicillin. Acid-fast, which reveals the presence or absence of mycolic acid, is important because mycolic acid is found in the cell wall of two genera in particular that contain important human pathogens. What are those genera? Put that into the chat. Yes. And thank you. If you weren't sure and just put a question mark or said you weren't sure, that's great. That's also an awesome contribution. Great. Yeah. So the two genera are the genus Mycobacterium. Now this has several species in it, one of which is the causative agent of tuberculosis, Mycobacterium tuberculosis, another one which causes leprosy, Mycobacterium leprae, and there's several others, Mycobacterium aquatica, which causes like a tuberculosis-like disease in fish, etc. And then nocardia, which can cause skin infections in people. So there's several species in this genus that are dangerous human pathogens. So if we suspect that someone has tuberculosis or leprosy, this is a very simple test to rule that in or out. Okay, now when it comes to special stains, what were the special stains? I'm going to have you, there were 3. I'm going to have you raise your hand and name the stain and tell me why we need a special procedure. Like, what about the thing that this special stain reveals makes it hard to stain otherwise? So raise your hand if you know one of the special stains and can tell me like why we need a special procedure. What's the reason we wouldn't normally see that structure, whatever it is. Jenica, go ahead.
Capsule stain.
The capsule stain, yes. Tell me why we need a special procedure for the capsule.
Okay, the common procedure darkens the background, so the capsule stands out as a clear area surrounding the cell.
Yes. And why is that necessary? Like, if you just did a simple stain, you wouldn't see the capsule. Why wouldn't you see it?
Because it's dark.
Well, you're on the right track. It's because it doesn't retain dye. Exactly. So it's that capsule isn't going to show up. But if you do stain the background in the cell, then it does show up as a halo around the cell, just like you're saying, because it doesn't take up dye. Yeah, exactly. Okay, great. Let's have another one. So we got flagella stain out the way. There's 2 more. Let's go hunting for those other structures. What else we want to stain, and why do we need this special procedure, guys? Tiffany, go ahead.
The flagella stain?
Yeah, why do we need a special procedure for that?
Because it's too thin to be seen.
Yeah, remember it's 15 nanometers, guys. That's way too small to be seen with a light microscope. So what does the flagella stain do, Tiffany? Like, how does it help us?
I think it was like a mordant to the coat of the flagella stain to
Yeah,
make it like thick to be seen. I'm not sure I remember too much of it. But
yes,
something like
you're
that.
absolutely right. Yeah, it's a combination of dye and mordant alternately applied. So dye, then mordant, dye, then mordant. What mordants do is thicken stain. So it's kind of like a mascara, right? It just thickens the flagella. And last but not least, what is the 3rd type of stain in the special stain category? And why do we need a special procedure to see it? What is that 3rd one? Go ahead, Tiffany.
I was actually trying to put the hand
Oh,
down.
you're trying to put your hand down.
It's
Okay.
gram stain, I guess.
Gram
Maybe
stain
gram
was
stain?
differential. It was differential.
okay.
That one's been covered. But thank you. I appreciate you. You answered the last one as well. So let's not put you on the hot seat too much. It looks like Jenica wants to answer. Let's give her a shot.
Endospore stain
Yes. And why do we need a special stain for that one?
stains endospores.
Yes. What about the endospore makes it difficult to see using any sort of like just a simple stain?
Because it does not like readily take up stains.
That's right. It doesn't readily take up dye. It's got a very tough spore coat, so the dye can't get inside. So we need a special procedure to see it. Good job, you guys. Okay. So again, the capsule stain, it's also called a negative stain. Capsule staining is used because the capsule is made of sugar and sugar doesn't retain dye. The flagella stain is used because the flagella is really skinny and it's way too small to be seen with a light microscope, but you can use dye to artificially thicken it, just the same way mascara thickens your eyelashes. And then finally, the endospore stain stains those endospores. They're inside the cell to protect the DNA, and the spore coat around the endospore is really, really thick and it resists dye. Okay, now, why do we care about these special stains? Why would we use staining to look for these things? Again, if you have an idea for any one of these, raise your hand and tell me, we want to know if there's a capsule because, or we want to know if there's a flagella because, or we want to know if there's an endospore because. What would be the reason we'd go do these diagnostic tests. Sarmeet, go ahead.
I guess the reason for that, so that we can differentiate them with the other microbes those are present in the slide, or maybe selected slide. So the ones do not take the stains easily. In order to differentiate that, we use different procedures: capsule, flagella, endospore, and that's how we get to know, we can categorize which microbe is present in which slide.
Yeah, these are useful for categorizing for sure, because these structures are not like, it's not like all bacteria can make them. And it's not like if they can, they just decide on Tuesday to make them and on Wednesday they don't. If a cell can make them, then it pretty much will make it and then it'll be there. And if it can't, it won't. And so that can help us to narrow down who we're dealing with. Any other reasons that you guys remember from your prep as to why you'd want to do one of these stains? Like think about what clinical relevance having this structure has to the organism in terms of our concerns about it being able to hurt us. Gabby says it can help us understand how to treat it. Sometimes that's true. I would say that's maybe like sort of a little bit lower on the list of reasons to do these types of stains. However, since it is part of the identification process in that sense, for sure, it's helpful in learning how to treat it. Any other thoughts? It is, yeah, so these are used to stain specific structures inside the cell, like capsules, flagella, and endospores. Let's talk about it for just a smidgen. From the prep, you might remember this, and when we go forward into chapter 4, you'll want to review this. Capsules are present only on some species of bacteria. And when they're present on a human pathogen, the capsule confers invisibility on that bacterium. So in your body, it's really hard for white blood cells to see it. Okay, so they can avoid being phagocytosed by your phagocytes. In other contexts, like things that aren't human pathogens, they use the capsule for other things. Okay, but if we're talking about human pathogens and when we're talking about clinical implications, we're really focusing on things that make us sick. Those bugs that have a capsule are harder for our bodies to clear. Our innate immune system is at a disadvantage. What is a flagellum for? What's the purpose or function of flagella, guys? Yeah. So motility for sure is an important thing to know about, and a motile bacterium could be more dangerous than a non-motile. There are a couple of other clinical reasons. Leslie, you had your hand up. What would you like to share?
Isn't it can enter other bacteria more quickly?
So yeah, having that flagella certainly allows it to move around more quickly and potentially get into different parts of your body. There is a type of motility that we'll talk about later when we get to chapter, which one is it? The pathogenesis section, way later in the quarter, where some bacteria can get into our cells through different methods. They're not using flagella in that case. The other thing, and so you'll want to go back and review the prep on this and also make sure you're paying attention when we learn about it again in chapter 4. But flagella are important in two ways as diagnostic tools. So one way is they can help us to look at the pattern of flagella, which is somewhat distinctive sometimes. And also the protein that makes up the flagella is antigenic. Your antibodies, you have antibodies that can be specifically made against different types of flagella. And for that reason, it's possible to use that as a marker of who a particular microbe is. So they're important for motility and also identification. And if we know we're dealing with something that is a human pathogen and we've discovered that it has flagella, so we know it's motile, we can then possibly use that information to help identify what it might be. Last but not least, what's the function of endospores, guys? What if you can make an endospore, what's the purpose for the bacteria from the bacteria's point of view? Yeah, Haley, it confers heat resistance. Is that all? I mean, we've talked historically about how endospores provide heat resistance, so that's definitely an important thing. Is that the only thing that an endospore is resistant to? So we'll revisit this again in chapter 4 next week. Exactly, Sarah. It can survive in many different harsh conditions, okay? And that includes antibiotic exposure. So if you have a pathogen that's causing an infection and you take antibiotics to clear it, and the antibiotics are great at clearing the vegetative cells, they might not destroy the endospores, in which case that's, then it could recur. The course of antibiotic treatment might not be long enough to kill all of the cells. Endospores allow cells to resist all kinds of conditions, including antibiotic exposure. All right, now we want to talk about procedures. So we need to know how to do these things. That was part of the curriculum for this section. So I'm going to give you a second to look up or remind yourself what the procedures are for all these different stains. I'm going to give you a few minutes, and then I'm going to cold call you to find out what you know. And remember, as always, you are welcome to pass if you can't. Like, let's suppose you're driving in a car right now and you're like, oh man, I can't look this up. And then I call on you. You can just say pass. I don't want you to have a car accident on my behalf, please. But take a second to look these up if you can, or review and be ready. Okay, now that you've had a chance to think about it, let's do a little cold calling and find out. And so I just want to remind you again, what I'm asking for is not like super hyper detailed, but just in general, what are the steps, or what is the basic procedure? For some of these, it's pretty simple, or it hasn't been well described. You would just sort of say something descriptive. But for the gram stain, acid fast stain, and the endospore stain, eventually you want to know the 2, 3, or 4 steps in each of these procedures, respectively, with the names of the reagents. So just as much as you can remember, like even if you're like, well, I know that a dye named blah, blah, blah is part of the procedure, that's all I know. Or I know that there's a heating step or whatever. So I'm going to cold call you and we'll just kind of go through these. Let us begin with Nikki. What do you remember about simple staining? What's involved?
I know you prepare the slide and use crystal violet.
Yeah, that's absolutely one way to do it. You prepare the slide and then you use a dye. It could be crystal violet. It could be anything, really. It could be methylene blue. It could be safranin. A dye, simple dye, and bloom. You just rinse it off and there you have your cells. Excellent. Okay. Next, I'm going to ask Lori for gram staining. If you're able to unmute, Lori, what are the steps in gram staining, or any of the details that you can remember? Lori's going to pass. No problem. Thank you. How about Amy? Amy, would you like to give us your answer or pass?
You would use a purple dye?
Mm-hmm. Absolutely, a purple dye. Yes. Does anyone have anything they want to add? Purple dye is absolutely key. If you know the name of the purple dye, or if you know any of the other steps. Sarah, what would you like to add to that?
For gram staining, I have that there's 4 steps written down. The first step is that the primary stain is added, which is crystal violet. And I also wrote down, based off that chart that you provided, that the first step will stain all the cells purple. The second step is using a mordant. So in this case, it's the iodine. And then it says the cells will remain purple with this step. The third step is adding alcohol, which is a decolorizer. So then the gram-positive cells stay purple and the gram-negative cells become colorless. And then the last step is adding safranin, I'm not sure how to pronounce
That's
that,
right.
which is a counter stain. And then that will make all the gram-positive cells will still stay purple, but then the gram-negative cells will look red or pink.
Yes, good. Okay, so just to sort of reiterate some of the ones we've talked about so far, the simple stain is just a single dye, rinse and observe. The gram stain, as was just described, is crystal violet first, which is that purple dye, then iodine, which is a mordant, then alcohol, which is a decolorizer, and safranin, which is the counter stain. Okay, now acid fast has 3 steps. If you remember any of the steps, you can share them with us, or you can always pass. That's completely fine. But let's go next to Angie and ask you if you know any of the steps in the acid fast stain.
All I know is that the stain that is commonly used is the methylene blue, and the carbofuscin.
Yes, those are the 2 major reagents, the 2 major dye reagents in this stain. Good job. So there's carbofuscin and there is methylene blue. Does anyone want to add anything to that great answer? Those are the 2 dye reagents. There's a 3rd reagent. What order do they come in? Anyone remember any of that stuff? Yeah, Caddy says the order is carbofuscin first. Yep, that's the red dye. Acid next, acid alcohol next, and methylene blue is the 3rd step. Good. Yes, that's right. So we have this hot pink or hot red dye. Not hot as in heated, but it's just red or pink, depending on your perspective. Then there's an acid rinse, and then a methylene blue is the counter stain. Okay, now for the capsule stain, you are not required to know all of the reagents, but just basically describe the procedure. And for this one, again, you can always pass if I call on you and you're not sure. I am going to ask Janelle if you know how to do this one.
All I know is it uses an India ink to stain the background.
Yeah, so there's 2 different methods. India ink is one of them for sure, and staining the background is absolutely crucial and critical to this stain. Does anyone want to add anything? I'll just wait for a raise your hand if anyone has anything they want to say before I kind of sort of throw my 2 cents into the mix. Maybe there's something you know that you want to share. That's a great question, Ayumi. For capsule staining, we don't typically need heat. That's a great question. There is a stain that does require it. Okay. So yeah, the capsule stain, remember, we're trying to dye something that doesn't dye. Like basically it does not take up dyes. So what we have to do is stain the background instead. One method is to use India ink. Another method is to use 2 different types of dyes. Either way, it's called a negative stain because it stains the background instead of the capsule. The flagella stain we've already described. Let's see if we remember it though. I'm going to, and no pressure if you don't, maybe you were like trying to soothe your 5-year-old who was having a total meltdown when we were talking about it. So I'm not judging you. Kelly, do you know how to do a flagella stain?
From what I know, I do know you have to use heat for this one. And I think you do malachite green for this one.
You are
and then
thinking about the endospore stain, and those are correct for the endospore stain. So yeah, you're just like reading my mind 1 step ahead of me, but yes, you're absolutely right. And there's another procedure, and we're going to talk about it in a second, that does require exactly those things. The malachite green and the heat, super critical for the endospore stain.
Do you want me to do flagella?
If you know it,
because I know that one.
Yeah.
I know you use the carbofuscin as like a simple stain, and then you do a mordant, and like you said, you use like the mascara kind of technique.
Yes, exactly. Yes, you're right. So this is the one where we alternate, like we take a dye and then a mordant and a dye and a mordant. We do it over and over, just the same way that you might like use mascara multiple times to get the desired effect. But often, as Kelly mentioned, the dye is often carbofuscin. It can be other things too. It doesn't have to just be that, but that's a common one that's used. And then a mordant that thickens it. And you do this over and over until it's thick enough to see. All right, Kelly also gave us a great head start with the endospore stain. She said that there was heat and malachite green as the first step. Anyone who remembers any of the other components of this stain, go ahead and raise your hand. Anything else you'd like to say, like what's the counter stain or anything like that? Leslie, go ahead.
Is this a counter stain?
I wasn't quite able to hear you, Leslie. I'm so sorry.
Is the counter stain pink by any chance?
The counter stain is safranin. Yeah. And I couldn't quite hear what you said. So if that's what you said, then you're right. All right.
Sorry.
Yeah, no worries. Thank you. Thank you for being brave and being willing to share your answer. I'm sorry that the audio was a little garbled, but that is the Zoom life, isn't it? We all have to just live with it. I'm sorry about that. But yeah, so the endospore stain has malachite green and steam. This is necessary to get dye into that really tough spore coat. That's why we need the steam heat. And then the safranin is the counter stain that's pink. Okay, now I'm going to show you some pictures and you're going to tell me if this is a positive result for that particular stain. Is this a gram positive? Yes or no? Excuse me. Wonderful. You're all hired as my technicians. The answer is yes, this is gram positive because it's purple. And purple is a tough thing, right? Because when people say purple in the civilian world, they mean a lot of different colors. So purple in the gram world means like dark purple. Some people have described it as black or blue even. So dark purple is positive. And then this lighter color would be a gram negative. Maybe there's some gram negative cells in the background. It's definitely positive. Okay, here is an acid fast stain. And the cells are obviously blue. So is this a positive result or is this a negative result? Put that into the chat. Great, this is a negative result. What color would you see if it was positive? Right, exactly right. You would see red if it was positive. So yay, this person does not have tuberculosis or leprosy or whatever the reason was you tested them to begin with. This is negative. Okay. All right, now I'm going to show you an image and you're going to try to tell me what type of stain was done to get that image. So here's an image. We got these pink rods. We've got little green circles. Some of them are, the green circles are around in the environment. Some of them seem to be inside the cells. What are we looking at? Yeah, exactly. This is an endospore stain. And again, how did we get this stain? We used malachite green and steam heat to get the green into those spores. And then we used the safranin counterstain to counterstain the cells. All right. Here's another image. What type of stain was done to get this image, do you think? Excellent. You are all correct. This is a negative stain that is showing us the capsules. So there it is. Okay, now I'm going to give you some scenarios. You're going to tell me what stain I should use. I just want to know if the bacterial sample I am about to stain is, if the morphology is cocci and if the arrangement is clustered. What's the best thing to do that's going to take me the least amount of time? Yeah, I think a simple stain would be great. Now, some of you said gram stain, and that's not wrong. That would in fact reveal this. It is just going to take you a little bit more time. So if we're feeling like efficient / lazy, we would just do a simple stain. What if we want to know if this person has an infection that can be treated with penicillin? Yeah, here is where the gram stain would be really useful. Okay, and if we get a purple cell, what is the answer? Will the penicillin work or not work? If the gram stain reveals a purple cell, will the penicillin work or not work? All right, so we have a 50-50. And so hopefully by the end of chapter 4 next week, you will feel like super solid in this answer. I'll give you kind of a heads up. Remember that the purple cells are gram. What are they, guys? Purple cells are gram. Yeah, positive. That's right. Most everyone remembers that purple cells are gram positive. And the gram positive purple cells are penicillin sensitive. We'll find out why that is next week. All right, what if we suspect someone has tuberculosis? We want to do a like really quick experiment to see if that's a possibility. Yeah, because the organism that causes tuberculosis has mycolic acid in its cell wall, then an acid testing would be great. And what will we see if we're right, unfortunately, for this poor person? What color of the cells will we see if we're correct? Yeah, they're going to be red. All right, now we're dealing with this situation where we want to know if a bacteria has a capsule or not have a capsule. What would be a good stain for that? Yeah, this one's easy, right? It's called the capsule stain. You could call it the negative stain too. That's also another word for it. Now, what if we suspected something had motility and we want to find out? What would be one way to approach that? Yeah, we could do a flagella stain. That'd be a good idea. Okie dokie. Okay, what if we have a species that doesn't seem to be killed by boiling? What might be a first step to try to understand what's going on? Yeah, we could do an endospore stain to see if the endospores are the heat resistant, because there's another reason, right? If Bacilli survive boiling, it's either that you suck at boiling things, or that they have endospores, or it could be that they're an extremophile and they like boiling water. You don't know what it is until you do this test. Okay, so I'm looking at the time and I've overshot our break accidentally. I'm so sorry. I meant to take a break about 7 minutes ago. You're probably like, please give me a break. We're going to take an 8-minute break so that we can come back at 6:15, but I need to give you a break right now. So 6:15, and I'll see you then. All right, guys, welcome back. So let's talk about it. These are applications that are designed to help us think logically about the procedure and what we know about how something is done so that we can really more deeply understand it. So let's suppose that you did the gram stain, okay, but you forgot the mordant step in the gram stain. What do you think the color of the cells would be at the end of the procedure? For this one, just put a color into the chat. You know what the two possible colors are in a gram stain? Well, actually there's 3, I guess. You forget the mordant. What color do you think it's going to be? Put a color into the chat. If you have no idea and you don't want to guess, go ahead and put a question mark, but put something into the chat. Great. So we basically have some question marks and some people, I would say 50-50 on the pink versus purple. Great. The answer in this case, the most likely answer would be pink. And if you can justify your answer, if that was the answer you chose, and it wasn't just a guess, which is also fine. I love it when people guess, and I encourage you to do that. But if you have an actual reason that you want to share, go ahead and raise your hand. Tell us why you said that. Why did you say pink? Kelly, go ahead.
I said purple and then I changed my mind to pink now, because when you wash away the stain, those that didn't get, stabilized and the stuff didn't get broken down and the purple is going to stay in the cells that need to stay purple, it's going to wash away and I'll be pink.
Yeah. Yeah. So if you did everything right in the gram stain except for you left out the mordant step, the mordant step's really the thing that sets the crystal violet in the gram-positive cells. Without that, when you apply the alcohol wash, it tends to decolorize everything. And then when you do the safranin step, everything turns pink at that point. Okay. So what if you did the gram stain again? Okay, but you're just having a bad day. You remembered the mordant this time, but whoops, you left out the alcohol decolorizer. What color do you think the cells would be at the end of the procedure now? Put that into the chat. Great. So in this case, the most likely outcome is purple cells. Regardless of the actual gram status, it's most likely going to be purple. Tell us if you know why you chose that answer. It's also fine if you just guessed, but if you didn't just guess, if you have an actual reason for choosing that, tell us what it was by raising your hand. Megan, go ahead.
The alcohol is the decolorizing solution, which removes the purple color from the gram-negative cells.
Yeah, exactly. So the whole purpose of the alcohol is it washes the purple out of the gram-negatives. It doesn't wash them out of the gram-positives. We'll see why that is when we get to our structures. But the alcohol wash decolorizes gram-negatives specifically so that they're clear. And then when you apply the pink safranin, they turn pink. If that step doesn't happen, then everybody stays purple. And then when you add the pink, it doesn't do anything, right? It's too light. So it'd be like trying to write, use a yellow crayon on top of a black, background. You're not going to see it. Okay, so color after gram stain of an acid-fast organism. What do you think, guys? Put your answer into the chat. You've got an acid-fast organism and you do a gram stain on it. What color will you see? Don't Google this. Think about it logically. Lots of good guesses. All right. Let a few more guesses roll in. If you have no idea, put a question mark. But if you have an idea, say what it is. All right. So the best answer so far is Kelly's answer. And she said, would it stay uncolored? And that, I think, is an excellent guess. Kelly, can you tell us why you said that?
I said that because the acid fast things that are acid fast, they have that waxy mycolic acid layer, and so they're not going to want to die at all.
Yep, that's exactly right. The whole reason that we have to do the acid fast test is that those organisms that happen to be positive for an acid fast stain are not going to be positive for anything else because they have that waxy coating, which resists normal dyes. And I would say resist in particular water soluble dyes. All the Graham reagents are water soluble. Now, if you look online and you ask Google, Google will say, oh, well, the structure of the cell wall is gram positive because they know this from doing like structural analyses, which you don't have access to. And it'll stain this, that, or the other thing. But that's really only because as cells age, the cell wall gets a little bit unstable. And so they start to get some pigment sneaking in. But if we had really healthy cells, which is what we would assume, then the most likely thing is we wouldn't see anything at all. Okay, if you did a Graham stain, could there be any circumstance where you look at the result of your Graham stain and you might say to yourself, I wonder if these cells make endospores? What kind of appearance might you see in a Graham stain that would suggest that to you? Write that into a chat. I love it. All your answers are excellent. Some of you are saying, I don't know. And that's a great answer. I don't know is a wonderful answer. I appreciate that answer. Some of you are saying, I have an idea. Here it is. Excellent. And it's also always fine if you're feeling a little unsure to read the chat stream and see if you're inspired by somebody else's ideas. See if what they say makes sense to you. Of course, I may always cold call you on your answer. So you want to only pick someone else's answer if you're like, oh, their answer makes you realize something. Okay, great. So many people put something useful in here. And I think the first person to say it was Haley. Haley said there would be clear spots in the cell. Haley, can you tell us more about that?
Yeah, so I was thinking about how those endospores don't take up dye. They're resistant to the dye without the right treatment. So on a Graham stain, they'd probably be clear.
Yeah. Endospores are at least initially generated internally inside the cell. And because that's the case and they are not easily stained, then if there's an endospore inside of a cell and you do a Graham stain, the most likely thing you're going to see is like a clear hole in the middle of the cell that didn't receive the Graham reagents. And that might make you think, either I suck at Graham staining today, physics doesn't work, or maybe there's endospores. Now, if I suspected there was endospores, what could I do next to try to verify that? I suspected the species had endospores. What could I do next to verify that in fact it does have endospores? Yeah, it's not a trick question, guys. The answer is very simple. You would be like, well, let me do an endospore stain. Now, to be very clear, you would not do the endospore stain on top of the Graham stain that you already have. You would go back to the species and say, let me take some more of you, put new cells on a slide, do endospore stain instead. Okay, let's suppose that you do an endospore stain, and at the end of it, the cells are pink. What is their Graham status? I need you to answer in the chat. I have 2 answers so far. Thank you for answering. Add your answer in, even if your answer is, I have no clue. That's an excellent answer. Great. Okay. All right, so most of you are saying gram negative, and I know why that is. Okay, you have gotten used to the idea of pink meaning gram negative, but it only means that in a gram stain. So this is I would say, you might think of it as a trick question. It's not really intended to be a trick question. It's intended to see if you, A, understand that pink is only informative in a gram stain, and B, can pay attention to detail. Because I didn't ask you, if I did a gram stain and the organism is pink, then what is its gram status? I did not ask you that. What I asked you is if I did an endospore stain and the cells are pink at the end, what's the gram status? So if I do an endospore stain, the counter stain is safran and the cells are always going to be pink. Will I know the gram status at the end of the endospore stain? Yes or no? Surmeet, question or comment?
I have a question. Your question is after counter stain, when you do the endosperm, are you asking that would you get to know it's endospores or not? Is that what your question is?
No, the question is, I did an endospore stain. At the end of the endospore stain, the cells were pink. What's the gram status of the organism? That was my question. And most of you said negative, and I said that's incorrect, though understandable. But it's incorrect because I didn't ask you what color are they at the end of the gram stain. I asked you if the cells were pink after an endospore stain, what's their gram status? So would it be unknown, says John. John, you are correct. It would be unknown. Haley, you're right. It would be unknown. John, tell us what made you think that? Like, where are you coming from there? That's absolutely on the right track. You still there, John? I think your audio cut out for a second. No, I think we lost him. Let's see if it's oh yes, yeah, sorry about that. Let's see who else. I think you're going to agree with what is said next. Haley, would you like to comment on why you said unknown?
Sure. So I was just looking back to that two-step for the endospore stain, and I know that the safranin causes, or that counter stain causes the other cells to go pink. And so that's why that would be the result, but not necessarily a gram negative status or anything like that.
Yep. Yeah. So, so this question is tricky, right? It is intended to trick you into saying negative because when you see the word pink and you're asked for gram status, your automatic tendency is going to go, oh, it's gram negative, because you know that pink is gram negative. But you have to be careful because this is not a gram stain that was performed. It was an endospore stain. And the pinkness has to do with the fact that there is a safranin step in this procedure that stains things pink always. It's not because they're gram positive or gram negative. They're just stained pink because it's the endospore stain and that's how it works. So it is important to kind of understand that. And it's also, here's the other reason I ask this question in this way. Your tendency when asked a question and in a class or in real life is to assume there is an answer and to try to provide that answer. And your brain kind of works this way too. So just remember that sometimes there is not an answer, right? The answer to this question, what's the gram status of an organism that is pink after the endospore stain? What's the answer, guys? Let's see if I have you in my page yet. You have to do the gram stain to find out. We don't know exactly, Kelly. We don't know. Sometimes the answer is we don't know, and sometimes we don't know is the right answer. I don't want you to feel afraid of we don't know, okay? Alrighty. So we have enough time for our microscopy Kahoot. It's fairly short-ish. Let's go in. I'm just gonna click out of here and go to our Kahoot. So open up your Kahoot, kahoot.it, and or open up your Kahoot app, depending. And make sure you can also still see my screen. I'm going to screen share in a second here. The pin is going to show up on your screen right now. So the pin number is 3689014. 3689014. Oh, Ayumi, while people are logging in, I'll answer Ayumi's great question in the chat here. Ayumi asked if I forgot the mordant. So if I forgot the mordant or the alcohol, what would the color be? So it's two separate questions. If you forget the mordant, then what'll happen is the stain won't set in the gram positives, and then it'll get washed out of everything, and then the counter stains of random will stain everything, so they'll be pink. If you forget the alcohol, which is the decolorizer for gram negatives, everything will be purple at the end. All right, let's get logged in so we can get rolling on this little Kahootski. Got 18 people on the call. We got 13 people logged into the Kahoot. We're getting closer and closer. See if we can get a little bit closer before we roll. Anyone else want to join in, hop on? Okay, we'll give it 15 more seconds and we'll get started. So again, the game pin is 3689014. Love that, House of Mouse. Okay, let's do it. If you need to, you can always join us in the chat, or if you're driving, don't just listen. All right, here we go. What morphology is shown in this picture? All right, the best answer is coccus because all I asked you was for morphology. Staphylococcus is a tempting answer, but this has a lot of information in it. We want to just the morphology, so coccus would be the answer. What is this arrangement? It is chained strepto. Again, streptobacillus, very tempting, but all I asked you for was arrangement, not arrangement and morphology. So just the arrangement. All right, here you have to type your answer if you can. It's showing you a picture. Type in the morphology seen in this image. Great. Yeah. So Vibrio, and there was one that was correct that just got accidentally had a typo. So you were right too. Vibrio, that little C-shaped morphology. To determine if an infection is Bacillus anthracis or Staphylococcus, both of which are gram positive, what would be the best thing to do? Now, I'm going to probably encounter some debate here. So let's see if y'all agree with my reasoning. This is not for points, guys, so don't get all sad. I chose a simple stain because these are both already known to be gram-positive. And if I know these are the only two options, then all I need to know is morphology and arrangement. So I could do a simple stain. If you were in the clinic, you'd probably do a gram stain just to make sure you were really right that it was gram-positive, okay? So these are kind of, might be answered differently on the ground versus in my theoretical universe. But in my world, we've been talking about how if all you need to know is morphology and arrangement, you don't need to know gram status because we already know these are both positive. There's no reason to do it. We just do a simple stain. Quick. Okay. Multi-select on this question, which is required for acid-fast staining. So it's going to give you some options for different reagents that might be involved in the acid-fast stain, which includes some of the acid-fast reagents. Doesn't have to include all of them, right? But includes some of them. Carbylfuchsin, lipid-soluble. Remember that acid-fast has mycolic acid in the cell wall. That's a waxy chemical. We need the carbylfuchsin dye because it's the only one that can penetrate through wax. It's lipid-soluble. Wax is a lipid. Great job. What does the acid-fast stain reveal? Yeah, it tells us if there's mycolic acid there or not. Good job. What type of stain is shown here? It is the negative stain. Good work. All right, multi-select. Which of the following is a feature of a capsule? This is a multi-select, so there could be more than one right answer. You pick them all. That's right. Does not absorb or retain dye. That's why we have to do a negative stain. Makes the bacteria invisible to phagocytes. True or false, this gram stain of patient's infection suggests that penicillin would be helpful. Now, you might not know what color these are. Okay, remember that in the pink versus purple debate, the purple would be really dark. These are pink cells. Right. Pink cells, gram negative, penicillin resistant, don't use it. What procedure was done to achieve this result? Yeah, it's a flagella stain. So we would apply dye, then mordant, then dye, then mordant, then dye, then mordant until we thickened it up. What procedure was done to achieve this result? Good work. It was the endospore stain, malachite green, heat, and safranin. You get to do one of those yourself in the lab. A patient with a severe cough gives a sputum sample and then an acid-fast stain is done. It looks like this. So what do we now know? Good. All right. So it was acid fast positive or negative, guys? Put that into the chat. Acid fast positive or negative? It was blue. So acid fast negative. So this person does not have tuberculosis. And we don't know if it'll be treatable with penicillin because we haven't done a gram stain. If they have coronavirus, which is certainly possible, we're not going to see it on this type of stain because it's too small. So what do we need to do? More testing. Again, never be afraid of that answer. You do a gram stain, but you forget the alcohol. What do you see? What color are the cells? You forget the alcohol. Whoops. Purple is right. Good work. The alcohol is the decolorizer. It removes the crystal violet from the gram negatives. Without the alcohol, that doesn't happen. And so we get purple, everybody. You do a gram stain on an acid-fast organism. What do you see? Yeah, most likely you're going to see clear as long as the cells are young and healthy. Yeah. Because that acid-fast organism has mycolic acid, a waxy lipid in its cell wall, which resists gram stain dyes. Last question is a multi-select. You do an endospore stain and see this result. It shows you what? Select all that apply. You do an endospore stain, you see this result. What does it tell you? Good job. Yes. The species can survive harsh conditions. That's what endospores do. It's clearly endospore positive. The species makes endospores. And then we really can't say anything about these other things because we have not done a gram stain. All right, let's see who made it to the podium today. I do know that if you didn't end up in the top spots, that you were just right behind everybody, hot on their heels. So good work. Okay. Let's go back to our slides. Just a couple more things to say to you. All right, so we have to talk about measuring cell size, and we're going to talk about it in lab. In the last 3 minutes of class, I'm just going to very briefly give you one example. Okay, so for lab 3 and for this lab 2 that you're about to do, you're going to measure cell size. And that involves having some kind of scale bar. And in this example, we're looking at a cell. We're trying to determine its size. Here's the scale bar it's provided. We just have to figure out how many of these cells will sit on this scale bar. In my example, I found 7. We take the scale bar, divide by 7, and we get a number that tells us the width of this cell. Okay, so that's all I'm going to say about it for now, but we are going to revisit this in lab with a lot of other examples. You're in the midst of your just getting ready to move forward on your unknown ID. You have a number now. Don't forget to get started on this project. So part 1 is due not this week, but next week. However, I highly recommend you do it as soon as you can. If you submit it early, the unknown ID check and submit it early, then you can just email me and I'll grade it sooner, and that will give you a chance to know what you should do. All right, so let me just sort of mention a couple things. Tonight, pre-lab is due, and you want to bring your procedures to lab 2. We're going to, even if you don't, maybe there aren't any procedures, it's pretty straightforward, so I wouldn't worry too much about it. Not for Wednesday, but next week. I don't know what I was smoking when I wrote that. Next week, we're going to talk about cells. So do the cells pre-quiz, do all the topic page stuff, cells, video lectures, slides. Next week, we're going to have prokaryotes on Monday and then eukaryotes on Tuesday. Now, I cannot emphasize this enough. Moving forward, we're going to pick up the pace. I hope you have not felt it was too overwhelmingly fast yet, because it's about to get faster. And I want to say it's going to be double time. It's super critically important that you come prepared to the Zooms. Do watch the lectures. You've got 5 days before we meet again in lecture to watch those video lectures, take your notes, fill out the study guide, try to understand it, and then come here. And I'm not saying that because you did awesome today, you guys. That's not it at all. I just want to prepare you to be really diligent about not falling behind, because I want all of you guys to succeed. It's always sad to me when people don't. So I'm just trying to give you as many tools and a heads up as possible. All right, so don't forget to come to lab tomorrow night at 5 o'clock on Shuckson Hall in the basement. And we're going to get rolling with some microscope stuff. Now, if you've got questions, stick around. If you don't, I'm going to stop the recording, let you log out. Have a wonderful night. Stay warm. It's cold out there.